Right. Thank you, everyone. Very good morning to everyone. And I would like to, start my presentation. And by the way, my title is "What's Your Missing Matter?" First of all, I'd like to thank you, the organizer, GSI, and also AWS, especially Charlie, for inviting me to present the topic here, "What's Your Missing Matter?" So my name is Mavis. I'm from Oxford Nanopore. I'm clinical sales specialist covering the region. So Oxford Nanopore is one of the AWS partner who provides a sequencing platform. And this platform actually enables you to be able to access to the genomics context in an unprecedented resolution and release what's your missing matter. And we know that in the past 20 years, we have witnessed that there are rapid changes in the methods that used to analyze the genome, from the very first Sanger sequencing to do the very first, human genome, to high throughput microarray, to then the next generation sequencing. And because of these projects, millions of samples have been genotypes and hundreds of thousands of samples have been sequenced. And we have indeed made a very significant progress in understanding the disease. However, despite all these efforts, we know that many common complex diseases remain very poorly understood. And now we know that apart from the SNVs and indels, there are also structural variants, copy number variations, and also epigenetics, which are previously inadequate covered by the traditional technology. However, these are found to be important as well and playing important roles in the mechanism of the diseases. And with the recent advancements in the long-read sequencing technology, such as Oxford Nanopore technologies, genomes are becoming more and more complete, and this has actually led to the building of a better reference genomes. For instance, 2 years ago, the Telomere-to-Telomere Consortium has actually published the first telomere-to-telomere genome, and it actually unlocks the remaining 8% of the genome that is previously inaccessible with the technology in the market at that time. And it is also very important to build human pangenome for a better representation of the genomic landscape across a diverse human population. And for that, I would like to quote that Michael at Waters, that the use of a reference genome that is more representative of the populations of a given patient will be more likely to accurately link the genomic variants to disease. And now let's take a look at how the nanopore sequencing works. So if you look at the image or the animation on the left-hand side, you can see that nanopore devices use the floor cells. And these floor cells, it contains many of these tiny holes here, what we call a nanopore. And this nanopore is actually embedded on an electro-resistant membrane. So this nanopore, each of this nanopore is actually correspond to its own electrodes and then connected to the channel and the sensor chip, which is used to measure the electric currents that flow through the nanopore. And during the sequencing, the electric current will be applied. And when the molecule passes through the nanopore, the current is then disrupted to produce a characteristic squiggles. As you can see from the image here, so this is a squiggles. And this squiggles is then decoded using the base calling algorithm to determine the DNA or RNA sequence in real time. And what are some of the benefits of the nanopore sequencing is that, first of all, direct sequencing of DNA and RNA. And we are the only platform that can do direct RNA sequencing, because a lot of the sequencer in the market they can do DNA sequencing, but for RNA you need to convert it to the cDNA first before doing the sequencing. But for nanopore, you can actually do a direct RNA sequencing, and the advantage is that you will be able to get that epigenetic information as well. So and secondly, because our workflow do not involve PCR, so there will be no amplification bias, and the workflow is also very simple. And although Nanopore is always known as the long-read sequencing platform, but the fact is that we are pretty read-length agnostic. We have customers that use Nanopore to do the cell-free DNA metalomes, because cell-free DNA are generally shorter, but you can use the Nanopore sequencing to do that purpose as well. Essentially, what we sequence is, in terms of the read length, will be what you provide. If you provide some, the shorter fragment, they will sequence provide you the sequence with short fragments reads. And if let's say you are able to provide a very long DNA fragments, then our sequencer will just sequence and give you that sequence. And we are also the only platform that can do real-time analysis. It means that you can actually set that you want to collect the data, and then you want to analyze it every 20 minutes, every half an hour, or every 1 hour until you get enough information for your research objective. You can actually stop the run, and you can store the floor cell, wash it, store the floor cell for your next experiment. So this is the kind of the flexibility that Nanopore sequencing can provide you. And it is also very scalable, because in Nanopore, our objective is to enable the analysis of anyone, anywhere, right? So we ensure that we have the offering from a single platform, a single floor cell platform that enable you to run a single floor cell in the field. Let's say, or maybe if you have a pilot study, you want to test out how it works, you can always use a lower throughput platform here, MinION, with one single floor cell, and all the way up to high throughput platform that you run 48 floor cells at a time, in representing 48 whole genome sequencing can be run simultaneously for our highest throughput platform. And annually, this platform can generate about 5,000 genomes. And very unique to our Nanopore sequencing is that we have these adaptive samplings. So because targeted sequencing is one of the most used application in the sequencing industry, so these adaptive samplings enable you to actually do targeted sequencing or even deplete the region of interest. So how it works is that you really only need to provide a bed file, and this bed file actually will contain your region of interest or the region that you do not want. So essentially, it's just the chromosome number and the chromosome position and what is the gene. And during the sequencing is that every single second when the DNA pass through the pore, we will sequence at 400 base pair per second. And then during that process, right, let's say every 1 second you get that 400 base pair of information, then we will do a real-time alignment to check whether this particular region or sequence is of your interest or not. If yes, then the sequencing will continue. If not, then the reverse potential will be applied, and then to eject these particular strains from the nanopore. So the objective here is that it then frees up the pore and then enable the next strand to go in, because the next strand that go in, it may be your target, it may not be your target, and then the same process begin over again. It actually reserves that sequencing capacity to sequence only the gene of your interest. And yeah, you can see that this image over here, you can see that over the times, right? This is an adaptive sampling for the MHC region, and then this enrich region, the coverage that increased over the time as compared to the background. So if you are working on, for example, you want to sequence, sometimes you want to sequence the pathogens in a horse and you do not want a horse genome, you can also use the same method to deplete that, saying that I do not want the horse genome. And I think the beauty of this application is that usually the targeted sequencing, because there's involvement of PCR, so the methylation data cannot be retained. But with adaptive sampling, you do targeted sequencing, but you also can retain the methylation data. And then in addition, because of this sequence and rejection, sequence and ejection, you can actually get a number of these short reads that is ranged between 400 to 700 base pair, and you can actually do low-pass whole genome sequencing. So, in summary, with nanopore sequencing technology, we enable you to look beyond the genome, create a diverse population representation, and investigate the disease causality through a more holistic approach. So now let's look further into how long reads opens up the era of looking beyond the SNVs. So you see from this image here, you can see that more than 50% of the structural variants are actually missed by short read sequencing. And there are two examples here that where long reads drive a new biological discovery. First is in the dark regions. So previously, the dark region is inaccessible by short read sequencing technology. However, with long read sequencing technology, you will be able to access to read this dark region. And why is it important? Is because these dark genes also have a non-mutation that is associated with human diseases. And also, the second application is structural variants. You can see that about 30% of all disease-causing variation is actually made up of variation that are larger than a single base pair substitution. So I think one of the common questions when it comes to PopGen or the large core study is that can I actually detect structural variant at scale? And the answer is yes. So this study from the team at DeCord has shows that on the over 3,600 of Icelander. So I think what is important here is also that the phenotype that looked at, right, were not rare disease, but hypercholesterolemia, which is like common diseases. And in this case, they detected, a deletion to the PCSK9 and its downstream effect on the LDL cholesterol level. So this is relevant to many cohorts of the biobanks because they are not only looking at rare disease but also trying to decipher complex but common disorder. And over the recent years, you can see that there are more and more large initiatives that started to use long-read sequencing to decipher disease causality. The reason is that many of these cases remain unsolved even if they use multiple ways trying to find answer, even whole genome sequencing with the short-read sequencing. So, for example, in Genomic England, they partner with Nanopore for a project to diagnose unsolved rare disease cases. So there's about 7,500 samples, and most of it have undergone whole genome sequencing on short-read platform, and most of these cases are unsolved. And also recently, there's over 20,000 samples projects by National Institute for Health and Care Research. That they are using Nanopore to study the psychiatry, common and rare diseases. And as mentioned earlier, right, the inclusion from the samples from a more diverse population will be beneficial to restore the balance of representations and to allow for new genomic discovery, because the genetic architectures between population is different. And if you look at this image here, you can see that about 80% of the data until recent year available in the public data set are from European participants. However, if you look at the global population, European only represents about 16% of the global populations. And most of these are actually from GWAS study, and therefore a lot of missing information. And even in recent years, there are more programs on short-read sequencing, but still there are 8% of the genome that's still missed by the short-read sequencing. So in summary, the data sets that we have right now are not representative of the human population and is biased towards one ancestry. So The Human Pangenome Reference Consortium, so they have recently published the first draft of pangenome, and in this first draft of the pangenome, it contains the nanopore data that helps to assemble this first pangenome, and they find that actually making a switch to use a pangenome mapping is actually not significantly more computational expensive, but it results in an average 34% reduction in the false positive and false negative compared to using the standard reference methods. And this is one of the example of the the very large scale genome project that is using long read sequencing, the Admiralty Genome Program. So they aim to establish a database that has access to a very comprehensive view of the genome and forward translation into health benefit in the region through a number of programs such as the newborn screening program, the rare disease program, the NCD, and cancer. And in this program, they actually sequence 5,000 genome in a week towards 100,000 of long read sequencing. And I think this is very relevant to us in many countries in the Southeast Asia, in Indonesia as well, because we do have a lot of diverse population, indigenous population. And in this study here, this is a study to capture the indigenous Australian genomics diversity because it's very underexplored. And you can see here from this study, they find that 73% of the large indel and structural variants are previously unannotated, and about 30% are actually exclusive to indigenous Australian. And why is this significant is that many of these are encoding sequences and likely to have a clinical phenotypes. And of course, we also want to point out that not all projects will start massive. In these studies, so the sample size is relatively small, about 400 individuals. However, in these 400 individuals, they have reported more than 100,000 of non-redundant variants, which over 45% were novel. So this is important because it shows you that how you cannot relate the finding from one population and apply it to another population. So you need to do a lot of the local population genetic studies. And closer to the clinic, you can see that in this example in Seattle Children's Hospital by Dr. Danny Miller, what they try is that they want to see whether or not they can use long read sequencing as a single test to replace all these sequential testing using multiple platform, multiple technology, and still give less than 50% of diagnostic yield for the case that they receive. And they also want to see that how much is the diagnostic yield, how much it increased with the additional information given by the long read sequencing platform. However, the challenge here is that how do you characterize and also prioritize a new variant if you don't know their frequency? So in order to do that, they actually start to do the 1,000 genome project using Nanopore platform in order to create that structural variants catalogs, and then also to expand the understanding of the variations in the difficult to analyze regions. So this is their very latest publication, just published like 2 months ago. And you can see that they reported a higher recall and precision for SNVs with Nanopore versus a short read, especially in the well-characterized genomic region. And because of the long read sequencing capability, we can also resolve complex allele. Like for example, in this example here, this is a CYP2D6 gene. It's a very challenging gene to be characterized by short read sequencing. You can see that for short read, it is difficult to do phasing. But with long read, it is very obvious that you can see haplotype 1 and haplotype 2. And this individual in the haplotype 1 can see there is this deletion over here. The CYP2D6 gene is being deleted. And the haplotype 2 actually carries a star 36 plus star 10, which is a hybrid tandem arrangement. This is very difficult to be detected by the short read sequencing, and also not directly being able to detect by microarray. However, this is very relevant to our Asian population. And of course, when we talk about complete genome, it also, complete information, it also means looking beyond just the genomes. And in recent years, you can see that in these few areas, especially aging, metabolics, neurological, and psychiatry, these are some of the areas that I find that more and more growing evidence to show that epigenetics is playing an important role. So I think the question here is that if when you do the genome, whole genome sequencing, and then if you can get free epigenetic information as well, will you say no? So in nanopore sequencing, it is really a click of the button for you to collect, the epigenetic information. You can just click this and to choose whether you want to collect that information. It comes with no additional cost and no change to the workflow, no additional library prep is required. And in these studies, right, because all this year when people want to do methylation studies, they will use array, and so this study actually they compare the correlation between the array versus the nanopore sequencing, and the author actually reported a high level of concordance between the technologies. However, when you use nanopore, you can also discover a new loci. And in this Nature Methods paper here, they actually, the author developed a nanopore-based protocol. This is actually a protocol that enables you to do haplotype, resolve variation, and methylation calling in one single platform. And what they find is that it is possible to achieve state-of-the-art small and structural calling performance using only nanopore. And they have reported the SNVs with the F1 score of better than short-read sequencing, and also in terms of the structural variance, the F1 score is comparable to the hi-fi sequencing. However, the advantage that's being highlighted is that this can be achieved with a lower cost and also a higher throughput. And in terms of the methylation, they find that the methylation core were very highly concordant compared to the standard bisulfite sequencing. However, in addition to that, with nanopore, because of the long reads, you can actually have the haplotype-specific resolution to identify differentially methylated regions. And you can see that in nanopore, I think this is very specific or exclusive to nanopore platform, where you can actually detect 5MC and 5HMC, and you can see that there are actually 9 possible methylation states when you look at 5MC and 5HMC. So you look at the image on the right hand side here, 5MC is in red color, and in pink color here, this is 5HMC. So with the additional information, it actually allows you to do more scientific discovery. So as compared to the legacy multi-omics, if you want to do, beyond genome, you want to do epigenetic or transcriptomics, right? You might require multiple libraries. Like for instance, you might need to use array, or you need to do bisulfite conversion, which is a totally different workflow compared to a standard whole genome sequencing workflow. However, with nanopore, you can just use one library prep, one workflow, and then you get all this information. It's more streamlined and straightforward. And in nanopore, we understand that it is important for us to have tested this streamlined workflow for you, so that you can spend less time on optimizing all these, the workflow, and focus more on the science. So we do have the library prep kits for different applications, and these kits actually comes with the overage that is ready for or compatible for the automation purpose in the lab. And of course, when it comes to sequencing, we do have different devices, different throughput that you can choose of, and I think this is being highlighted in this today's event quite a lot, which is the data analysis. So we know the importance of data analysis, especially with the level of the informations that can be generated from nanopore platform, and so we are we actually have the different workflow for different application, whereby it is also available in the very simple graphical user interface where you can just click it. So it is available in the local version where you can install in your workstation or high performing computer, or you can actually do it on cloud. And this is how the workflow, looks like. Like for instance, if you want to investigate human variation, after you do the sequencing in real time, the methylation data is recorded, and the alignment can happen real time during the during the sequencing, or you can actually do alignment after the sequencing run. And then this data will be loaded onto our Epitome, which is where we host all the different workflow that you can select from. And one of these workflow is called WF human variation. So this is a workflow that is most used by the researcher that is doing investigation of the human variations. So this workflow here enable you to obtain the annotation files and reports for small variants like SNP indels, but also copy number variations, STR expansions, and also phasing. And at the end, you can actually visualize your annotated genomes with a methylation signal here. And you can actually do it directly on our Epitome, or you can actually use your own preferred genome browser. And these slides here summarize, what are some of the workflow that is available currently, and what is the GPU and CPU intensity required for each of these workflow or process. So you can see that during the base calling, right, it requires intensive GPU. However, when it comes to analysis, it's more on the CPU side. And the CPU intensity, it really depends on the complexity of the workflow that you are running. If let's say you are doing 16S or bacterial assembly, then it might not require to use a very high CPU intensity. But however, if let's say you are doing like human variation workflow, whole genome sequencing, and then you can imagine that we not just only analyze SNP and indels, but also copy number, STR, expansion, etc. So it requires more CPU intensity. So here, AWS has been playing a very crucial role in the sequencing world, especially in the population scale projects. This is in order to address that computational intensity for the massive amount of data generated from the sequencer every single day, be it like the base calling or alignment or analysis, or after all these projects finish, the huge amount of data that is needs to be stored. So I think one of the example that how Nanopore works with AWS is the collaboration with G42 for the Emirati Genome Program. So I think this is also the program that we mentioned earlier, and is one of the largest sequencing, long-read sequencing projects run by Oxford Nanopore and supported by the AWS platform as well to process this 100,000 long-read sequencing data. So I really encourage you to read the benchmarking of the Nanopore best scholar on AWS. This is what they do. And I think this is good because they do a lot of comparison of performance, and they also do analysis on the runtime and the computing costs for the Nanopore best scoring, and also they give you very detailed information on the test procedure and architectures. And in summary, right, you should really consider an architecture for base calling that allow you to choose the right compute environment depending on the requirement for base calling time and cost. So what they find here is that number one, a setting that requires processing a number of samples in short time will benefit from high-performing instance. And on the other hand, a larger population scale genome research project will benefit from the cost effectiveness of the smaller instance types. So I think if you do have, like for example, if you have if you are doing product developments and you want to put your pipeline onto cloud for scalable implementation across the world, you can talk to AWS experts over here. And then of course, if you have cohorts projects, you have large projects, and then feel free to talk to the experts here in AWS. And if you have a project that and you wish to actually look at more comprehensive genomic insights, and then feel free to reach out to Nanopore and the GSI here as our distributor. They'll be able to assist you and then with your projects. Yeah, with that, thank you.